Hi, this is Dr. Neela Bokore, and today we are going to start with microbodies. Microbodies are spherical structures which were discovered by Rodin. And he observed these tiny structures in the kidney tubule cells of mouse. These spherical structures are single membrane bound, that there is only one layer. So, one layer or one membrane which is similar to the basic plasma membrane, phospholipid protein bilayer structure, and inside there is a matrix. This matrix mainly comprises of enzymes, and this matrix is in the middle, which is called the core, and a membrane. So, basic spherical structures with a single membrane and matrix which is rich in enzymes. These microbodies, they do not arise from ER or Golgi body. In case of lysosomes and all, we talked about that these structures, they pinch off from Golgi body or ER. They do not pinch off. So, not formed from ER or Golgi. So, then how are they formed? They are actually formed by assembly of proteins and phospholipids which are present in the cytoplasm itself. So, those proteins and phospholipids of cytoplasm assemble to form the membrane and the structure. They assemble to form these microbodies. There are three main microbodies that we talk of. Let us take the first one. These are known as they are found in animals only. We are talking about animal peroxisomes. So, this is the first microbody, and as the name tells us, they are found only in animals, eukaryotic cells of animals. So, the cells where these peroxisomes are found are kidney cells or cells of kidney, liver, testis, adrenal cortex, brain, and even in the cells of intestine. So, intestinal cells also have these peroxisomes. Structure is same, spherical with a single membrane. These peroxisomes which are found in animals, they have two main types of enzymes. One enzyme is hydrogen peroxide synthesizing enzyme or producing enzyme. This enzyme, as the name tells us, it is going to produce hydrogen peroxide. It is known as oxidase. So, the enzyme which is helping in production of hydrogen peroxide is oxidase. The second enzyme which is present in animal peroxisome is hydrogen peroxide degrading enzyme. Degrading is breakdown. And this enzyme is known as catalase. So there are two main enzymes. Now what do these two enzymes do? The substances which are in the cell, they are first converted into hydrogen peroxide. Amino acids, uric acid, they are converted into hydrogen peroxide, that is H2O2, in presence of the enzyme called oxidase. This hydrogen peroxide, as soon as it is formed, it is going to help in killing the cells. So this actually kills microbes. So this is a part which is helpful for the cell because it is acting as a disinfectant. But hydrogen peroxide, if it is accumulated in the cell for a longer period of time, it acts as a toxin. So immediately it should be degraded. So this hydrogen peroxide is broken down into water and molecular oxygen. The enzyme which is going to help here is catalase. So these same two enzymes, that is oxidase and catalase, are helping in formation of hydrogen peroxide and then breakdown of that same hydrogen peroxide. So all these substances which are not useful anymore, they are converted into a substance. This substance for a short period of time can be useful, but after that short period it becomes toxic, so it needs to undergo degradation. It is converted into water and molecular oxygen. This molecular oxygen will be available for oxidation reactions. So now these two enzymes are related with peroxide, and so the name given to this microbody, peroxisomes. One more important role which is played by peroxisomes in case of animals, especially in human beings, is detoxification of alcohol. People who consume alcohol, 50% of that alcohol, only 50%, other 50% does not get detoxified. It remains as alcohol and then causes the damages to the body. 50% of alcohol, from this 50%, hydrogen is removed. So from 50% of alcohol, hydrogen will be removed and is given to molecular oxygen to form water molecule. Water is a harmless substance, on the contrary it is required by our body. So this 50% of alcohol, after losing this hydrogen, changes into a harmless substance. So 50% of alcohol gets detoxified by these animal peroxisomes. Let us take another microbody. It is called glyoxysome. Glyoxysomes, they are found in germinating seeds. That means they are of plant origin. They are present only in plants. The enzymes which are present in glyoxysomes help in the conversion of fat or oil, which is in the form of stored food, in case of these germinating seeds, into sucrose. So till the seed actually germinates and starts producing its own carbohydrate, till that time all that reserve fat or oil which is present will be converted into sucrose, and this sucrose will be made available to those seeds for performing their metabolic activities. So all the enzymes which are required here, they are present in glyoxysomes. The reactions here which take place are through a cycle which is known as glyoxylate cycle, and all the enzymes which are required for this are present in glyoxysomes. So glyoxysomes are found in germinating seeds. Now once this seed has germinated, it has its own root arising from radicle, plumule giving rise to the stem, and the leaves. Once the leaves start performing photosynthesis, dependence on this sucrose is over. So after that, these glyoxysomes will also disappear. That means glyoxysomes are present in plants only in germinating seeds. Once the germination process is over and the plant is on its own, then these glyoxysomes disappear. Next microbody, third microbody, same peroxisomes but which are found in plants. So they are called plant peroxisomes. Plant peroxisomes are found in green leaves which are performing photosynthesis. Green leaves performing photosynthesis. Those leaves will have these peroxisomes which we call the plant peroxisome. Structure is same. The enzymes which are present in plant peroxisomes are combination of the enzymes which are in animal peroxisomes as well as in glyoxysomes. So the enzymes which are here are same as animal, the same as the ones which are found in animal peroxisomes and glyoxysomes. Plus, they also help in some more reactions. Glycolic acid is oxidized because they have this oxidase also, same as animal peroxisomes. So in presence of this oxidase enzyme, this glycolic acid is converted into glyoxylic acid. It is oxidized. Glycolic acid is oxidized into glyoxic acid. This glyoxic acid can change into succinic acid, and succinic acid will then produce an amino acid called glycine. Now here I said this can change into succinic acid. Can word is used because this is one alternative pathway. So there is other pathway also. So this glyoxic acid can take part into that pathway also. It may change into succinic acid also. Succinic acid can participate. Again we are saying can because this is one alternative pathway. Succinic acid can take part in Krebs cycle, and glycine can take part in protein synthesis. It's an amino acid, so it can take part in protein synthesis. Now for all these conversions, enzymes would be required. So here also there are enzymes which are required. This conversion will also have enzyme. Conversion of glycine for protein synthesis and succinic acid in Krebs cycle. All these enzymes are present in plant peroxisomes. So it's a complex structure. Complex in the sense there are so many enzymes. There are oxidases, there are catalases, plus there are these glyoxylate cycle enzyme, plus there are enzymes which would help in conversion of succinic acid into Krebs cycle and then glycine to protein synthesis. That is why it is considered as, this plant peroxisome is considered as the most complex microbody. So there are three microbodies. One is a peroxisome. Because they are found in animals and plants, we have divided it in two categories, animal peroxisomes and plant peroxisomes, and glyoxysomes. Animal peroxisomes only in animals, plant peroxisomes only in plants, and glyoxysomes only in germinating seeds. After germination is over, they are lost from those cells. So these three are the microbodies. Hello again. Now we are taking a major organelle which is found in plant cells only, and that is plastid or plastids. Plastids are exclusively seen in plant cells and some photosynthetic protists. So they are specially related with the plants and some photosynthetic protists. Plastids are double membraned structures and they are of three main categories, that means types. The types are basically on the basis of what is present inside those plastids. So first type are known as leucoplasts, which are colorless plastids. Second, chromoplasts, which are colored plastids. In some books, you may find three terms given: leucoplast, chromoplast and chloroplast. Chloroplast is the green pigment containing plastid. So when we are saying chromoplast, these are pigment containing plastids, that's why they are colored. So we are talking about chloroplast as a part of a chromoplast. So if the pigment is green, then they would be called chloroplast. The green pigment which is present in chloroplast is chlorophyll. We will take chloroplast structure in detail. If there is a red pigment present, then those are known as rhodoplast. The pigment is red. Some pigments are carotenoids, leucopene or leucopine, which is found in red pepper or red chilies, plus phycoerythrin. This is also red pigment. So any of these pigments can be found in these plastids, because of which they will have red color or they will impart red color to that structure. They are called rhodoplast. The third is known as pheoplast, which contain yellow-brown pigments. Yellow-brown pigments like xanthophylls. So depending upon which colored pigments they have, chromoplasts can be green colored or red colored or yellowish brown colored. So that is why we have kept chloroplast here, but in some books, as I said earlier, they have divided the lipids into 3 categories, because chloroplast is a very important plastic. That's why they have given a third name here. So they have said leucoplast, colorless, chromoplast, colored, and chloroplast, green. But when we are using pigment, then it should become a part of this. That's why we are taking 2 types of leucoplasts. All plastids, sorry, not leucoplasts, 2 types of plastids. All plastids, all types of plastids, they arise from proplastids. And these plastids can change from one form to another. For example, in a green unripe mango, the outer cover is green. It looks green because it has more of chloroplast, more of green pigment. When these mangoes, for example, when they ripen, the color changes to orangish or yellowish. So that is when chloroplasts are converted into the other colored pigments plastid. It could be rhodoplast, as when they have a yellowish pigment. It could be the pheoplast, but it is chloroplast which is getting converted either into rhodoplast or pheoplast. So they are interconvertible, but all of them, they arise from structures called proplastids. Let us talk about the leucoplasts also. Leucoplasts are colorless and they help in storage. So they are also known as storage plastids. What do they store? Would give us different types of leucoplasts. If they store starch, then they are known as amyloplasts. Amyloplast, colorless, no pigment, they are meant for storage and storage of starch. If they store proteins, then they are known as proteoplasts. There is one more term given to these protein storing leucoplast, is based on the protein which it stores. So protoplast would store protein, and if that stored protein is alluron protein, which is most commonly that one, so then they are also known as alluroplast. Alluron is that protein which they store. So if it is any other protein, it would be just proteoplast, but most commonly, as we said, the protein which is stored is alluron protein, so they are also known as alluroplast. You can also write it as alluronplast. And the third, which store fat or lipids, then they are known as ileoplast. As lipid is being stored, there is another name given to it, is lipidoplast, based on what is stored inside. So leucoplasts are of 3 types depending upon the stored material that they have. It can be starch, it can be protein, it can be fat. Starch storing are amyloplast, protein storing are proteoplast. There is one more term given on the basis of the main protein, and ileoplast, fat storing or lipid storing. So 3 types of leucoplast. Chromoplast are 3, but there is one more pigment which is present in plants, and we have not given any specific name for that pigment containing chromoplast. That pigment is a purple colored pigment, anthocyanin. This anthocyanin, purple colored, when is present, then it is simply called chromoplast. Like here we have a name for green red and brown, we don't have a name for this purple colored pigment. So if a colored plastid has this pigment, then we will simply call it chromoplast. Let us take the structure of chloroplast. This is the most important plastid which we will be talking about in details. The reason is very obvious, that it helps in photosynthesis. So let us take the detailed structure of chloroplast now.